Welcome everyone to Corpus Curiosum, the fifth series of this lecture, and we turned 2 years actually this year, this summer. So we were very glad to sort of be on this journey for 2 years already with you guys. We're stimulating critical thinking in neuroscience, and our mission is to aim to stimulate critical thinking in neuroscience through engaging with early career researchers. That's us, but it's also you guys, and fostering discussion, really sort of connecting different fields together within neuroscience, different schools of thoughts, and these kind of connections, they kind of mirror the corpus callosum in the brain, sort of connecting the left and the right hemisphere. And similarly, we want to connect different parts of the neuroscience world together to create these kinds of synergies within the field. So just a little bit about our values, like what we stand for, and sort of like what we want to push sort of going forwards with these lecture series. We want to support early career researchers, which is something that's very important to us, enabling collaboration and creating a supportive environment at eye level where like everyone feels welcome, everyone can connect in this kind of sort of sometimes very, very hectic world of academia, where it's sometimes quite hard to find a voice in these kind of very rigid structures. At the same time, we want to embrace diversity, fighting intersectional inequalities, be it racism, be it sexism, be it sort of other forms of discrimination, and amplifying underrepresented voices. It doesn't only mean giving underrepresented voices in our talks a space, a sphere to like communicate their ideas, but also, of course, to sort of engage with researchers from different parts of the world, since the Western sort of research landscape is quite dominant. And lastly, to be accessible. So, as you noticed, all our talks are free. And sort of like in our quest for like advocating for open science, we definitely want to lead by example, and being accessible and being free to access online, of course, makes it easy for all of us to come together from different parts of the world, no one having to fly in. And this is sort of like a nice legacy that we carried over from the pandemic. Our sponsors are the International Brain Region Brain Research Organization, IBRO, and FENS, the Federation of European Neuroscience Societies, which very generously supported us in this series, and which we were very proud to have as backup. These kind of big organizations supporting this endeavor. And from the IBRO, we were awarded for the second time consecutively the Diversity Grant, which sort of like reflects our our ambition and our work, and sort of like bringing in diversity in neuroscience. So this is something that we will work on, sort of moving through this kind of series, which we will also see in future talks. A little bit about us. So we are Ines, Faisal, and Alba. We are from different places in our career, different universities. We are spread across the UK, Spain, and Switzerland. And sort of we're trying to sort of like bring you guys together and sort of see this diversity also within our team. And another big part of our endeavor is to sort of kind of get the community together and to give you guys a space as well to contribute. And we're very, very happy and very, very lucky to have so many ambassadors for different countries, from different countries, representing Europe, North America, South America, Asia, and Africa. We have our amazing community manager, Mariela Trinquero, as well, who is helping us bring all this community together. And many of the faces that you can see in the picture, you will also see in today's talk, and I've already spotted some. And we're very, very grateful to have you in our ambassador program. If that's something that's interesting to you, definitely reach out to us. We'll be having similar things coming up in the future. And then about you as guests to this talk, I mean, we have attendees from over 45 different countries in the world. You can see here in this world map. And this again shows, how mixed we are as a group. So, take the chance, reach out, you have the chance to like, connect to people, to researchers in Brazil, Argentina, and India, Nigeria, so forth. So you can really sort of see this platform as an international way to like collaborate with people who are like-minded and also early career researchers. And yeah, now sort of moving on to our speaker, and we're very, very happy to have Folu Oyefeso with us. He is a PhD candidate in the Biomedical Engineering Science Department at Loma Linda University. At this institution, his lab primarily studies stressors found in the space environment, which can be harmful to astronaut health during long-duration mission. Folu plans to use his background in bioengineering to answer questions about how the brain is affected by these stressors, specifically space flight radiation. As the brain is exposed to these stressors, it speeds up the aging process, which may contribute to neurological disease and dysfunction. Thus, Folu is determined to answer scientific questions in brain aging and use insights from these studies to inform future strategies to tissue regeneration in the central nervous system. And today we'll hear a talk from Folu on what can we learn from tiny brains, big lessons from organoid culture.
Okay, great. So I've been really excited to give this talk today. As mentioned, I work in the PICO lab at Loma Linda University. I'm using brain organoids to address some of the questions about the effects of spaceflight on astronaut health. Right now, it's sort of a short-term, 6 months, maybe a year in the space station, and then coming back to Earth. But as we start to think about spaceflight and some of the implications of exploring other planets, we have to think about the long-term effects of that. I'm not going to spend a ton of time talking about my research today, but I'm definitely open to answer any questions. I'm going to kind of leave it to some of the greats in organoid research and give you guys a background over what organoids are. So we're going to look at this question in really 4 stages. First part is about developing brain organoids. So what's a little bit of the background and how are people culturing these organoids? There's now quite a few different methods for culturing them. Next, I'm going to cover some of the useful applications for brain organoids. So the 2 main things that people look at are development and disease. And then after that, we're going to go into some of the ethical implications of brain organoids. So I'm going to present a couple of themes, and then we're going to discuss sort of what those issues are. And then after that, it's going to be an open discussion. So let's just get right into it. Okay, so some of you may know, if you've worked with stem cells or have heard some things about them, that the use of embryonic stem cells for research has been a controversial issue, and that's usually due to the public perception. And also there have been some things historically where the research needs to be carried out with greater scrutiny, or they needed to visit the ethics before starting the research. And so because of that, scientists tried to think of other ways that they could explore stem cell research without using embryonic stem cells, which would be derived from blastocysts. And so to basically move that needle a little bit, some of you may know the name Shinya Yamanaka and also Takahashi. They published a study in which they used basically embryonic and adult mouse fibroblasts to then generate induced pluripotent stem cells. And so some of the differences between the stem cells that you might think about in the body and induced pluripotent stem cells, the embryonic stem cells are generally considered totipotent. They can become any cell type in the body, versus induced pluripotent stem cells. Pluripotency just refers to becoming, they can differentiate into cells that follow one of the 3 germlines. Typically, we think about ectoderm, mesoderm, and endoderm. And so in 2006, they published the first study where they generated those stem cells from mouse fibroblast cultures, which was a really foundational study for organoid research. And then they decided to investigate that even further. And in 2007, they published a follow-up paper in which they generated these pluripotent stem cells from human fibroblast cultures. And so that's moving from mice to human. And so if you look at sort of, I found a great graphic which kind of gives a good overview of these factors. These are transcriptional factors which you can induce in the cells, you can cause them to be overexpressed, and then that creates a stem cell-like state in these previously more mature fibroblasts. And so they call them the OKSM factors, which would be OCT3 or 4, SOX2, CMYC, and KLF4. And so you can use those factors, and this is using a retroviral approach. So it's kind of like you have a plasmid, and it's like a cassette, and then you can include the part of the gene that you want the cell to express. So you expose the cells to that retroviral plasmid, and then the cells start to express those factors. And so using this stepwise approach, they looked at what's the perfect combination we can use to express these factors and then end up with induced pluripotent stem cells. So this was a very technically sound paper. So because we're using stem cells today, that's how you know that they took a very solid approach and it's very reproducible. But they started with CMYC, and they saw that introducing the cells to CMYC can cause the chromatin to become uncoiled. So that's a process that has to do with acetylation of histones, basically the DNA being wrapped up, and also methylation. And so when you have acetylation, it actually sort of unwinds the DNA that's wrapped around the histones. And so that makes it more accessible. And so they saw by using CMYC, they could kind of uncoil the chromatin. But if you don't add additional factors, then you can see the cells start to go down an apoptotic or senescent pathway, or they could become like tumor cells. And so you have to add an additional factor. So the factor they added was OCT3, and KLF4. And so that becomes sort of an embryonic stem-like cell. But then adding the additional factor SOX2 is what allows the cells to become induced pluripotent stem cells. And so this is, once again, a very foundational approach that they use. And so because of this research, we're now able to use these cells for many, many different kinds of research. And so I just included the paper title there. But what's most important for our talk today is the fact that because of this research, now we can use these induced pluripotent stem cells for organoid research. And so I tried to go back as much as I could and do due diligence to some of the founders of this kind of research. And Yoshika Sasai is one of the teams that really started this development of cortical-like, 3D structures. And so this is a step up from using the traditional 2D culture, where it's flat and it's a monolayer culture. And though those studies are really good and we can learn a lot of things about the specific effects of drugs on different cells or the differentiation of those cells, what you're really missing is that 3D architecture, right? And so when you think about animals, animal models are great because they have that 3D architecture. And so you can see a brain, and some of the features are similar to the human brain. And then we have some, homology between the different genes. So we can study those, and then we can sort of carry out research using the mouse models and translate them to humans. But what's missing in those animal models even is that human component. So they do have some humanized animal models, but there are still genetic differences between animals and humans. And so that's why organoids really shine through as a good approach to identify how genes and, let's say, proteins are changing in cells that you would find in the human brain or human body. And then you can, as far as I'm convinced, but I'm going to try and convince you today, that because of that, you can try and translate these, these technologies, these methods to humans. And ideally, we want to see greater efficacy of these treatment approaches. So I personally am interested in neurodegenerative diseases. A lot of those neurodegenerative diseases have a basis of increased oxidative stress. The cells become stressed and they start to age faster. It's a really rudimentary way of saying it, but that's the general idea. And so it's cool because using this model, we can actually start to address some of those diseases using human cells. So what this team did was they had embryonic stem cells, and they sort of like introduced those clumps of stem cells into this pseudo extracellular matrix called matrigel. And once you put them inside, the cells start to aggregate together, and then you can add additional factors to the media. And some of the patterning factors, that's what they call them, because you want to pattern these cells to become like a more mature cell type. And so they used FGF, fibroblast growth factor, they used Wnt and BMP, you guys know bone morphogenic protein. And by having this combination of different factors that you add, you can actually guide the organoids towards, whatever differentiation you want them to become. And there's not such a precise control when you're working in a dish. So the cells are receiving signals, and then, it's kind of like saying, oh, we're in the brain environment because we're receiving these signals, so we should become like brain cells. And if you were using a different cell type or different factors, let's say intestine, you could actually generate intestinal organoids or even heart organoids. So I'm extremely biased towards brain organoids. I think this research is incredible and awesome. I think this is really the next step for how we can treat human disease. But what they did was they used these factors, and then what they saw is that by adding these factors to organoids, which were cultured in nature dill, which were cultured in media under suspension culture, which means they were basically just floating in the dish, they were actually able to generate the same kind of cells you would expect to see in the brain. But even more than that, they were able to see some sort of low-level architecture. And so, cells have the ability to organize and migrate. And so what they saw is that there was this formation of these sort of like cortical-like regions. If you look towards the bottom, I don't have my pointer up, but if you look towards the bottom, if you look in, let's say, M, N, and O, or if you look at P, Q, and R, you can see that that has a really nicely defined sort of layer, you call it like laminar layer, where the cells have self-organized and ended up in those regions. So that's, I think it's probably unexpected for the people when they first started this research, but it's really cool to see that this is something that you can use in the dish. And right now I'm trying to avoid using the word recapitulate, but a good way to spot anyone who's doing brain organoid research is how many times they use the word recapitulate. So I'm just gonna say resembles a brain-like region. So you can see that this is some pretty well-defined structure just for being a clump of cells in a dish. And so moving on to 2013, this is really when that term cerebral organoid was started to, or they've essentially coined the term cerebral organoids. And so the difference in this study, which was done by Noblick and Lancaster's team, is that they use IPS, so induced pluripotent stem cells, versus Sasai's team, which used embryonic stem cells, but also they use induced pluripotent stem cells. So when you look at this study, you see that they kind of took a similar approach where they culture first a 2D layer of cells, then they allow those cells to become clumped in usually like a cup-shaped dish or a well. Then after that, they implanted them in matrigel. So you can see that's what that, right under it says day 11. It looks like they're injecting the cells into this matrigel. Then after that, they use this really cool approach called a bioreactor culture. And there are different ways where you can suspend the organoids in media, but with a bioreactor, it's kind of like a vat, and then it has this propeller that spins around. It circulates the media, and then it allows the structures to float around in that media. And so you might ask, why is it important that these cultures are suspended? And so the reason for that is that if they're not suspended, usually they can deform, along the surface that they're cultured on. So if they're on a flat surface, they're going to become more flat. And so if they're spinning, what they do is they become, for the most part, spherical. When you look at figure C, that's not a sphere, but they can kind of, once the cells start to grow, some regions grow faster than others. And so in this case, you see that the morphology overall is not really spherical, but there are still regions where you see that same kind of organization that we saw in the last picture. I just realized that there are a couple of typos on this, so we'll ignore those for now. But what they showed in this study is that you could generate these organoids from iPSCs, and then you can pattern them to become like brain cells. And then after that, you could actually use them to study certain diseases. So in this case, they actually use these organoids to model microcephaly, which comes from a loss or mutation of the CDK5-RAP2 and the expression of that protein. And so they showed that organoids that had a loss or mutation of the CDK5-RAP2 were actually smaller than organoids that didn't. And so this is sort of an early approach, but it does show that, with changes in genes that are known to have effects in humans, we can actually see some of those same things being modeled in organoids in the dish. And since that time, there have been many different organoid models that have developed. And so some of the most popular ones you'll see are cerebral cortex, hypothalamus, telencephalon, optic cup, anterior pituitary, cerebellum, hippocampus, and the list goes on and on. And so as this research starts to grow and the methods become a lot more streamlined, there's a really great potential to study the effects of certain stressors or drugs on specific brain regions. So what are some of the ways that people are culturing these organoids? So far you've seen where you can put them inside Matrigel and you can suspend them. Say if you have a shaker plate, you can actually have that in an incubator. So the organoids, these clumps of cells are in that shaker plate, and it's just going around in a circle and spinning and keeping those organoids suspended. You also saw you can put them in the Matrigel, but you can actually culture them without Matrigel. And there are actually some other approaches too that people have considered. For example, the slice culture. So one thing that happens with organoids is that as they grow in size, the center of the organoid starts to become, let's say, a little more necrotic because it doesn't have as much access to the nutrients that you'd see in the media, right? So if you think about treating a tumor, one thing you have to think about is how close is that tumor to the vasculature, because that's going to affect the treatment. So it's a little bit similar here, even though these are considered normal tissue or normal cells. As they grow bigger and bigger, there aren't any blood vessels in these organoids. And so the media can't get through and the nutrients can't get through the center of the organoid as well. So what happens is you start to see death within the center of the organoids as they grow bigger. So this example shows day 70 versus day 150. And so you can actually culture these organoids out. I think some people have done 200 days or 300 days. You can culture them for a long time, assuming you have really great culture technique. But what happens is that you start to have necrosis in the center. So one way to address that problem is actually by slicing the organoid. So you can slice the organoid, whether it's on the top or bottom or multiple slices, and that creates a sort of flat spherical-like structure. And you can culture that in a dish. So this is where you wouldn't be doing suspension culture anymore. But one benefit of using this is that you can actually see the development of more mature cell types. Because as the cells, throughout the structure have better access to nutrients, they're able to mature in a more healthy way. And so then you can see, better maturation in the organoids, even though you lose that spherical structure. So that's just one approach that has been used. And you can culture these in the media, but there are also some interesting differences if you culture them at what's called the air-liquid interface. And I'm going to avoid getting too details because I can hear myself kind of diving in too deep. But basically, when you culture them in media versus sort of at the air-liquid interface where you have them exposed to air, you can actually see differences in the cell types that pop up. And if you look on the right, this is actually one of my favorite cultures, and it says slice culture at the top. This is actually a microfluidic culture or a chip culture. So basically there's sort of a plastic surface and a membrane on top, and you can culture organoids in those chips. And so this is actually a relatively new approach for culturing organoids and cells in general. And I think this is really where the field is starting to move forward, but there's still a lot of development that needs to happen for organoids. But I thought this study is really cool because they saw that as you grow the organoid in sort of a thin chip, what happens is it actually starts to show this folding. So what you see in the human brain is that as it gets bigger, there's sort of a space limitation. That's why we don't have giant balloon heads. But to avoid the brain and the head continuing to grow larger and larger, there's this cortical folding. And so that it basically increases the surface area and allows more neurons to pop up. And so they saw that by growing these organoids in this thin layer, they actually saw some folding in the regions of the organoids, which is surprisingly something hard to do when you just have the spherical culture. So this is just showing that there's different approaches you can take. But in general, the organoid refers to something that's a 3D structure. And if it's a brain organoid, it has cell types you expect to see in the human brain. So there's very cool stuff there. But let's think more about some of these applications. So a couple of the applications for generating brain organoids. You can see that it's been used to model microcephaly. That was the second study I showed you. But other diseases and disorders have been examined as well. So lissencephaly is sort of a disease you see or disorder you see where the brain grows, but it doesn't have those folds. So the brain is smooth. And because of that, you actually have a reduced cognitive potential of, let's say, a developing child. And so this is a disorder that has not been well addressed because it's really difficult to understand what's going on in that process, but there are some genes that are expected to play a role. And so some researchers researchers actually used organoids and were able to model lissencephaly. So looking at folded brains versus brains that are just smooth. There's also autism spectrum disorders. So this is actually a particular interest of one of my collaborators, Dr. Alison Watry. And he's interested in these autism spectrum disorders because I think he actually has a family member who experienced autism. And so he's using this model to understand more about how autism affects the brain and if it has any effects on electrical activity. You can also see some other things like neonatal hypoxic injury and Down syndrome, which are diseases and disorders that really we have a hard time addressing in the field. You can also look at some things like infectious diseases. So Zika virus was one of the earlier viruses that was tested using brain organoids. And what it does is exposure to a neonate or an infant can actually lead to a reduced brain size. So you have small brain or microcephaly. And they saw that by exposing organoids to Zika virus, they actually saw the same effect of microcephaly, where the organoids were showing up smaller than the controls. You can also look at cerebral malaria. And then my particular interest was the neurodegenerative disorders. And so this area here is a little bit harder to address because once again, there are some limitations with growing out the organoids for a long time. And so many people would argue that these organoids are most useful for modeling development, but not as great for modeling mature cells. And so we have to be a little humble and say that even though there are mature cells in the organoids, there's still a process of defining what a mature organoid looks like. But they have seen some features of different diseases that they can model in brain organoids, for example, Alzheimer's disease and Parkinson's disease. So you can generate organoids to have dopaminergic neurons, which are the neurons that play sort of the biggest role in those diseases. And they saw that by doing, basically introducing genes or mutations that leads to those diseases in humans. They saw some of the similar features in organoids in a dish. And so this is just to say that there were a lot of different approaches that we can use these organoids for in a lot of different diseases that we can model. And it's only getting better from here as the method starts to improve and the culture techniques start to get better. And so now to switch gears a little bit. So I actually included a study, this is also done by my collaborator, Dr. Montry. And in this study, they showed that well, first, let me define a little bit. They generated organoids and then they put them on these MEAs, which are multi-electrode arrays. And if you think about like electrophysiology, basically that device has these electrodes and it's picking up signals from neurons. And so if you have two nodes that are next to each other, let's say there's electrical activity at one, and then you see the next one light up right afterwards. That's a way of saying there's like a transmission of electrical activity across the organoid. And so this is really interesting because they were able to show that once these organoids were cultured on the MEAs, they started to have more and more complex oscillatory activity. So that means, one side of the organoid is activated and then the other side of the organoid is activated. So it's sort of like a wave of electrical activity. Now, if you think about that, well, first, they then took those organoids and then compared them to neonates and saw that there was a surprising similarity between, let's just say, overall electrical activity in organoids and early neonates. And it does fall off after a period of time. For example, the neonate brain is obviously going to have more and more complex electrical activity, but then it does lead us into some important questions about the ethics of brain organoids. And that's something that we have to consider at every step. So if you think about these brain organoids, as they become more complex, we start to get into questions of, well, how complex does an organoid or any tissue have to be to really be just like the human brain, right? And so some people would argue that electrical activity, as it increases and it shows that there's communication, is sort of a low-level consciousness, but so far research has not suggested that that's the case. But you can see even when you're culturing, let's say 2D cultures, there is still electrical activity that propagates across the dish. So this is a question that's important to answer. And now we can dive a little bit into the ethical implications of brain organoid research. And so I was trying to think about the best way to approach this, but there was a study done by Hyun Sharpdeering and Lunshaw. I probably butchered those names, but I thought this was a really great study for kind of addressing each of these themes in turn. So theme one, so one of the biggest things to consider is what are the like the regulatory policies that affect brain organoid research. And so this one, I know I'm kind of getting close to the time, so I'll just try and go through them briefly, but there are different groups that you have to consider when you're thinking about how can we answer the question of, you know, are these brain organoids conscious? How far is too far? And sort of what's the consciousness or moral status of these organoids? So in terms of regulatory policy, we know that the public opinion shapes a lot of how research progresses, and it's really important to consider the ethics of research at every stage. And one of the biggest ways to ensure that the public is well informed is to really emphasize science communication. So if you think about stem cells and how that research was carried out early on, because the science wasn't communicated as well to the public, and then the papers are published, and then there's a lot of sort of, let's say, like BuzzFeed type articles with big headlines that get people really excited. That can lead to a lot of confusion and a lot of negative opinion and negative sentiment about the research, sort of pushing the borders of manipulating nature too much. And so I think it's really important to get the public involved at every step. And it's also really important to get regulatory bodies and ethicists involved early on in this research. So that way we can make sure the research is progressing without breaking any sort of moral or social codes, because those do play a big role in research. The other part of that is guidelines. So if you're doing any stem cell research, you will have your institutional committee for the use of stem cells, or if you think about ICUC for Animal Care and Use Committee. It's really important to have those committees involved early on so that the institution is aware of the research, and also so that the research can progress and the community is well informed, and also the research is carried out in a very, let's say, like high level and considerate manner. The next part of that is procurement of human biological materials. So anytime you use samples from a patient, it's really important that they're informed about what those samples will be used for. And so if you think about Henrietta Lacks would be one great case of where the cells were used, but then there was no restitution to the family, and the cells were used forever in longevity without having the right approvals early on. So if you think about collecting someone's stem cells or creating stem cells from fibroblasts, you might define one purpose that those cells would be used for. But this paper actually highlights a potential need to inform the patient once again if you intend to use those cells for brain organoid research. And the reason for that is that, consent in one area doesn't necessarily imply consent in another area. So it's really important that they're aware throughout the entire process. One of the challenges with that is that a lot of the stem cell and organ donations can be anonymous. And so if you think about that, let's say researchers carry out a study and then they find that in the cell line there was, a certain risk for a certain genetic disorder. There might be a need to inform that patient that they have a risk for this disease, and that could actually, improve their quality of life or prevent disease. But if the samples are anonymous, then there might not be a way to communicate with that patient. So there are a couple of questions that you can dive into there. But if you discover something in a dish, is it required that you go back and then tell the patient, and that might actually improve their quality of life down the line. The next thing would be translational delivery. So if you think about stem cells and early implantations, I think retina was one of the earliest stem cell implants. And you can see that because you have these pluripotent stem cells, there is the potential for those cells to become any cell type in the body in one tissue, which you don't want. Because I think most people would agree that having hair growing out of your eye is a bad thing. And so you want to avoid tumor formation. And so now, because there's been a lot more involvement of ethicists, and there's basically a lot more stringent process to use stem cells and definitely to use them for translational delivery. Some of those issues are not as prevalent, but they are always worth considering. So you do have to think about immune rejection. You do have to think about tumor formation. And so these are really important questions when it comes to the translational approaches of organoids. The next thing would be animal research. So One of the benefits of using organoids is that they have the potential to reduce the number of animals used in research. And so overall, you have less loss of life in that matter. But there are some methods that combine organoids and animals, and they call them chimeras. And so with that research, so far, what they've been able to do is implant, let's say, a brain organoid into a region of a mouse brain. And one of the benefits of doing that is that you can have the vasculature from the mouse brain grow into the organoid. And so then you get, let's say, like a more mature development of the organoid because now it has, some of the nutrients it needs. But then people start to ask the question, is that now a human mouse brain, or does the organoid have a brain of its own, or is the mouse going to become smarter? And so far, there's no evidence to suggest that those things happen outside of there is some signaling communication between the organoid and the mouse brain. And also there's the vasculature that starts to grow into the organoid to reduce some of the oxidative stress. But once again, it is a really important question. So even though the animals, were not seen to become sort of a mojo jojo where they, become a lot smarter, there was still an ethical question of, is this, appropriate research to do if we haven't really figured out what consciousness is yet? And the last thing is, of course, consciousness and moral status. And to repeat myself, we don't really know exactly what consciousness is yet or how to measure that or define it. Most people would take the approach of going through stages. So first, like, is there electrical communication? And then after that, is there sort of like a regional, like a wide scale communication, and also let's say the awareness of the environment. So is there input? Is there sense, like an input and an output response? And then after that, the question would be, well, is this organoid self-aware? And so I think at this point, we would be at, where there's signals which are received and then the cells can act on those signals. But there really is no evidence, and I would argue even a good way to study if there's consciousness in, basically a clump of cells. But what you can see is that an organoid is essentially a clump of cells and it doesn't have all of the brain regions intact. So it's not, a full brain region, let alone, a full body. And so I think we're very far away from being at a point where that becomes a very major concern. But I would agree that it's really important to look at that question early on. And maybe this would actually stimulate more science and research about, how do we better define consciousness? Because there are some really important implications of that for patients who are in the hospital and considered medically brain dead. So this research definitely is fast paced, and there are a lot of different methods and approaches that are being taken. And it's really important that we involve, let's say, ethicists, the FDA for drug testing approaches, and the schools, guiding bodies, the institutional committees for stem cells and other research to make sure that this research progresses in a way that has good science, but is also, it allows us to really inform the public about what this research is doing and what it's not doing. With that, those were some of the main points.